Initiating Cell Special Project. Experiment 1. Sweet Insight Yeast Respiration Rate across different sugar solution.
For yeast respiration rate across different sugar solutions, it means which sugar solution give the highest respiration rate by assess the hydrogenase using method blue and assess yeast respiration rate using spectrophotometer. Sugar with highest yeast respiration rate will decolorise methylene blue and produce most bubble in 4.5 cm and an average absolute value rate of 0.0368 every minute.
Introduction for yeast respiration is respiration in the process where energy is released through the oxidation of carbohydrates. It happens in mitochondria of plants, animals and humans. The aerobic respiration chemical formula is the sugar plus oxygen equal to carbon dioxide, water and energy. Our hypothesis is the monosaccharide sugar yield higher respiration rate than disaccharide sugars. Disaccharide sugars cannot be oxidized by yeast by respiration.
Experiment has two parts. First part being the hydrogenicity test and second being observable value of respiration rate. Okay, for first part, first step is to each test tube was labeled and filled with different sugar solution. A drop of methylene blue and yeast suspension was added and shaped to mix. Lastly, after 30 minutes, decolorization and bubble production was recorded. Second part, first step, spectrophotometer warm up, blend solution was prepared, inserted in moment. A mixture of sugar, BTB, yeast mix was prepared and initial value recorded and next reading taken after 30 seconds.
For the result of absorbent respiration rate, we are able to plot all the data into a graph and obtain the average absorbent value rate, with glucose being the highest, followed by sucrose, while galactose with an insignificant value. As for dehydrogenase activity test, both glucose and sucrose show a reaction of bubbles production and decolorization of methylene blue, with glucose leading. From this, we are able to calculate the respiration rate, and while galactose and distilled water show no reaction.
From this experiment, we concluded that yeast respiration rate is the highest in glucose solution. Next, glucose has higher yeast respiration rate than sucrose. Other than that, galactose has no reaction due to absence of enzyme to convert into glucose. And lastly, these findings is useful for efficient production of an industry.
Experiment 1 completed. Experiment 2. Live now living cells react to redox.
And for the redox reaction, we examine the effects of redox reaction to living cell by observing oxidation rate towards different treatment, comparing catalyst activity and exploring the effect of temperature and catalyst activity. For the experiment, we know the cells that are exposed to air will exhibit the most rapid oxidation effect. Liver or meat demonstrated the highest catalyst activity yield and increase in catalyst activity were associated with elevated temperatures.
Introduction for living cell react to redox. Enzymatic browning is responsible for the browning effect on foods facilitated by polyphenol oxidase PPO to produce quinones, which is brown pigments from polyphenol in cells. Catalyst is an important enzyme in living cells, probably for breaking down hydrogen peroxide into water and oxygen for cellular protection. Catalyst optimal temperature is 37 degrees, and like any other enzyme, temperature deviation affects efficiency.
Compartment potato and apple were placed into different wash bay and then treated with different treatment and then decoloration observed after 30 minutes. Next, for part 2, each test tube were filled with chicken liver, potato, soap peas and apple. Then hydrogen peroxide is added to the test tube and then the amount of bubble produced was observed. Next, for part 3, 3 slices of potato was treated with different temperature and then potato was mashed into test tube filled with hydrogen peroxide and amount of bubble produced was observed.
For oxidation treatment tests, both apple and potato slices show a clear discoloration in air, slight discoloration in water, and little to no discoloration in lime juice. In catalyst activity in living tissue, chicken liver produced the most bubbles, indicating high catalyst activity, followed by potato, soaked peas, and apple in decreasing order. Lastly, catalyst activity in different temperature resulted with low catalyst activity at 4 and 55 degrees Celsius, while high catalyst activity at 24 degrees Celsius.
From this experiment, we concluded that discoloration of potato and apple slices were induced by oxygen. Next, citric acid in lime inhibits polyphenol oxidase, causing no discoloration. Other than that, higher catalyst concentration yields higher catalyst activity, important in cellular protection. Moreover, catalyst activity optimal temperature is at 37 degrees Celsius, room temperature closest. These findings is important in food preservation and medical purposes.
Experiment 2 completed. Initiating end of presentation protocols. Thank you for listening. May this presentation benefited all of you. End of presentation. Goodbye. Initiating system shutdown.